So I'm taking here kind of like a bridge between what we were just heard about the curation interface and then the query and reporting interface. And I'll just start with a brief introduction on how we essentially got from IEDB to CEDAR for the query and reporting interfaces. So as Matt has been showing you, this is the basic data structure that we have. References like papers that describe epitopes, often multiple ones, that are then tested in multiple different assays that come with other factors. So as that enables us to essentially then query for data within what I'm showing you here is the IEDB that identify the types of references, epitopes, and assays that match this. So in the middle here of the IEDB homepage, you see kind of the primary query interface, which is like the most frequently accessed filters essentially on the data. And then you can go in the IEDB, as we were saying, it's like infectious agents as one of the key things. You can say, I want to look for things that are derived, epitopes that are derived from Pseudomonas, which is a bacterial pathogen. And when you search for that, these are the kinds of results that you're getting. You get this tabbed format where you're seeing the epitopes, and this is the tab that is visible here, all the epitopes listed that are derived in Pseudomonas in the IEDB, the antigens that contain them, the assays in which they were shown to be responsive, the receptors, so this is BCRs and TCRs, BCRs obviously being antibodies when they're secreted, and then the papers where these are originally described in. So that is the whole concept of the query mechanism, that you're defining your query, which filters the overall record, and you're getting this tab format back where you have different results. So essentially, these 422 epitopes are described in these 94 references. The same references might describe other epitopes, say from a different pathogen or so. But this is kind of the overall results in each of these tabs that match this. As Matt was showing, these tables are kind of giving you the high-level summary, again, of the kind of features of the data that are most commonly utilized. So in this case, looking at the assays, you have kind of a high-level summary that is here kind of written out. But when you click on these numbers, you're getting the detailed data that is actually captured. So you have the detailed information about the paper that is described here, the epitope that was utilized, and then things like this specific mouse strain that was used, it's a TCR transgenic in this case, specific comments from the curator that entered it or so. So like a really high level of detailed information. So then the idea is that as you're going in, ideally we always recommend to start with a very broad query, the broadest thing they're interested in, and then you filter down. So you can, for example, go to the query that we just had and say, not just Pseudomonas, I actually just want to see things that are where the host responding is human, and I only want to see T-cell responses. I didn't want to see B-cell responses. And you click this again, and you're getting the much smaller set of data back that matches your more strict criteria. So this is the IEDB data structure, and CEDAR essentially shares the exact same underlying data structure, but it was expanded to the specific to have a more accurate representation of the kinds of information that is important in cancer, like the tumor type, stage, and things like that. And that's actually where I'm going to hand it over to Zeynep, who's going to talk about how we took this basic data structure and expanded it specifically to make it relevant for cancer. Zeynep?
Thank you, Bjoern. Okay, let's see. Can you see my slides? Okay, great. So as Bjoern mentioned, we started off of our experience from designing the IEDB, but when we wanted to design the CEDAR website, we reached out actually to cancer researchers to understand their needs. So we conducted interviews with them, and we found out which fields were most critical for their work. And based on this feedback, we then introduced several new cancer-specific curation rules that we needed to accurately capture the cancer data. For example, we started capturing the cancer types in more detail. For example, we now distinguishing if it's a non-small cell lung cancer instead of just saying it's a lung cancer. And this applies to also other cancer types. We have now also introduced cancer stages. These can be very important when we're conducting immunotherapy studies, for example. So we're also distinguishing between how the tested host got the cancer. So if it's a naturally occurring cancer, if it's an animal model with cancer, or if there was a therapeutic vaccination. And speaking of vaccinations, we also distinguish between prophylactic and therapeutic vaccinations because it's a vastly different context in the cancer research. And altogether, our goal was to create a search interface that enables both intuitive and granular queries, while still providing a lot of flexibility for different research needs. So Bjoern just showed you the IEDB website. This is the CEDAR website. As you can see, it's very highly similar. We changed the branding a little bit to make it CEDAR specific, and we also introduced a few new panels, which I want to talk about now. So we have the epitope panel here, which we also have in the IEDB. Here you can search for any epitope. You can paste in your specific epitope sequence. You can also select if you want to use BLAST to look for partial matches and epitopes that mismatch your query epitope, for example. And then we have the epitope source. Here you can type in your gene name, your protein name, or also like a UniProt ID. This will help you also with an autocomplete to find the gene and protein you want to look for. And here we have a list of our different antigen types, including neoantigens, viral antigens, and the germline self or host antigens. Here we have another checkbox. If you check this one, the results will include other epitopes from the same paper, even if they're not cancer associated. So for example, if you might get data from like a control experiment, like when they used like influenza peptides in the same study, they tested a cancer epitope. In this panel, you can select the host. As you mentioned before, the curation is primarily focused on human data, but we also have some mouse and non-human primate data in CEDAR. Here we have the assay panel where you can select between T cell, B cell, and MHC ligand assays. And here again, in this text box, you can even search for a specific assay type you want to search for. This is a new panel that we introduced because the receptor sequences are getting highly relevant now with the new technologies. You can check these boxes to only include results that include TCR, so T cell receptors sequences, or BCR, B cell receptor sequences, and you can even specify which chain you want to focus on, or even a specific CDR3 region. Here we can select the MHC restriction, class 1 or class 2, or even specify a specific HLA allele down to the 4-digit resolution. And finally, we have the cancer panel where we can, again, type in here the cancer type. We can choose the cancer stage and also the exposure types that I've mentioned before, if it's a naturally occurring disease, an animal model, or a vaccination. We also provide these finders. So you can see in these panels, these finder icons. For example, here we have the assay finder, which helps you to find a specific assay type. You can use these text boxes to search for a specific assay type, or you can also click through the tree to find the assay type you're interested in. And we have the same similar construct for finding antigens and proteins, hosts, MHC types, and also cancer types. So this is also a great place to start to explore what we have available in CEDAR. So now we covered the basics of CEDAR, and I want to actually go into some use case examples to demonstrate what kind of searches you can perform using this database. In the first example, let's say you're working on a literature review about prostate cancer, and you want to find all studies that tested epitopes from prostate-specific antigen, also called PSA, and you're also interested in finding out which regions of the PSA protein are most immunogenic. So let's build the query for this. Here we can see I started typing PSA here, which gives you all the hits here in our among all the proteins that we have in CEDAR. And here the first hit actually is the prostate-specific antigen. And by clicking on this, you can select it. And next, we want to go into the assay panel and just leave all the assay types selected and also include positive and negative assays, for example. And because in this example, we want to know everything that has been tested. Yeah, after we select these two, we can click search. And then we get to the results page. Bjoern mentioned the different tabs that we have available. So let's start looking at the epitope tab. We can see on top of here, the current filters are highlighted here. So we're looking for the epitopes from the antigen PSA, and we're including both positive and negative assays. And we can see here we have 128 assays available. And here we can see the number of references that reported the specific epitope and the number of assays where this epitope was tested in. And each row is a unique peptide, and you can click on any of them on the CEDAR ID to go to the experimental details, which I will show you in a minute. So going to the antigen tab, we can see in this example, we have two antigens. We have the PSA antigen here, which has the most epitopes. But in this example, we also have a new antigen and prostate-specific antigen mentioned here. So this is the mutated version. So in the example, we wanted to know where the epitopes are located in the PSA. And for that, we can click on this bar chart icon here, which launches the Immunome browser. The Immunome browser shows me where the immune responses occur along the PSA protein. So here on the x-axis is the length of the PSA protein. And here, this top panel shows the response frequency. And then on the bottom, we see the number of assays for each position. And the positives are shown in blue and the negatives in red. And for example, in this example, we can see here this specific region seems to be highly tested. We have almost 40 positive epitopes and also some negative epitopes. And you can click on here to explore these regions in more detail. And there's also the table of the specific epitopes if you scroll down on this page, which I will not show in this presentation for the sake of time. So finally, for our literature review in this example, we can go to the references tab. And then here we have the 84 references listed. So this is a great place to start, for example, for performing literature review. You can download these results again by clicking this export results icon up here. And this brings up this pop-up here where you can select which fields of this tab you want to download. And then you get a nice, and select an Excel table or CSV table for better parsing, for example. Yeah, you can get a nice Excel table with all the details about the references, and you can look up them. So for our next example, let's say the researcher is studying recurrent mutations in cancer, and they use prediction tools, for example, to identify potential neoepitopes. They found one peptide that stands out and is recurring in multiple patients, and it was predicted to be immunogenic. But now the next question for the user is, has this epitope already been tested in the lab, or did they find something new? So for that, we take the specific epitope sequence, and then we can put it in here, and we leave it at an exact match. And then we check both positive and negative, again, because we want to see if the peptide has already been studied and it was negative. That's also important to know in this context. And then we click search, and then we get the result. And in fact, this epitope is a mutated epitope, which is also highlighted by this mute colon, and then it says where the mutation is located along the protein. So in this case, the protein catenin beta 1 is mutated at the position 30, where the S mutated to an F. And this epitope actually has been already reported in 7 references and has 12 assays reported in CEDAR. So it's not new, but this might be still interesting to see what's going on with this epitope. And for that, we want to click on the CEDAR ID, as I mentioned before, to go to the epitope details page. So on the epitope details page, we see a brief text summary here on top that describes how many epitopes, how many references. So basically what we saw before in text form again for the user to review. And then here, because in this case, we have a mutated peptide, a newer peptide, we have the variant information as well in this table here. So we have information about the gene symbol, about the protein and the protein mutation, also the DNA mutation on the chromosome level. And importantly, we also have the external resources linked here. For example, here we have a DB-SNP ID, so it was also reported in DB-SNP. So this can be reviewed by using this ID. We also include the damage predictions here from external resources. And in this tool, we include 12 tools, and then 4 of the 12 tools are predicted to be damaging, for example. And we also have the ClinVar annotations here. And ClinVar, it was also reported to be likely pathogenic. And then these are the cancer types in which context it was reported. So this is just a high-level overview. If you want more details, we can click on this Open Cravat summary. So Open Cravat is a collaborator of us. They have actually a fantastic tool where they use thousands of different resources and annotators for mutations. So this is very useful. If we click on this, they actually created this specific variant report for us. So I just screenshot at the top of it, but you can scroll down here and we have a lot of information from all the different resources. So this is very valuable when we are exploring a specific mutation. So going back to the epitope details page, and when we scroll a little bit further down, and we see this table here, curated experimental data, and we can see here all the assays that were performed and what the assays were. So we can see here, for example, under T-cell assays, we see 8 experiments, including cytotoxicity and interferon gamma release. And here we can see how many assays were conducted and how many of them were positive. And we also have MHC ligand assays. And reviewing this data, we can say this gives us strong experimental support that the epitope is both presented on common HLA alleles, and it's also immunogenic based on these assay types and their results being positive, most of them being positive. So one more example I want to show here is about viral antigens, because viral antigens can be quite interesting in the context of cancer, because they are recognized as being foreign. So in this example, the user wants to find viral epitopes that have been tested in head and neck cancer, and specifically those epitopes that had positive T-cell assay results. So to build this query again, we can start here in the epitope source. We are deselecting new antigens and the germline antigens, and we are only keeping the viral antigens because that's what we're interested in. And then we deselect B-cell and MHC ligand, only leave the T-cell. And in this case, we're only interested in the positive outcomes. And then down here, we can start typing the head to get to the head and neck cancer, and then select the corresponding entry, which is the first one in this case. And then once we hit search, we again get our results. And so this lists all the different viral epitopes associated with head and neck cancer, which we use as a filter. And in this case, all of them are derived from alpha papillomavirus 9, which is consistent with these HPV-associated head and neck cancers. So we can now also switch to the assays tab to see exactly how these epitopes were tested. So this tab includes details about the method. So here's the assay description and what the outcome of the assay was. Also, the MHC restriction is also reported here, and the specific antigen that was used in the assay. So this is actually the tab that has the most of the details among all of the tabs. So this is actually also what I like to export if I want the highest level of detail. Again, so this export result is available on all the different tabs. So once we click here, we can also see again in this assay tab, there's many more fields available that we can export. And these are also expandable by clicking on the plus sign. So the user can just leave it as is and download all the information, or to make it more readable, you can just customize and select the fields that you're interested in. So there's actually multiple ways to access the CEDAR data. I just showed you how to use the website, which is quite intuitive, hopefully, as I had shown you. But we also provide bulk downloads, which I will show you in a second. And we are also currently working on ways to access the data programmatically. Our API is almost ready and should be released in the coming months. And you also just received funding to build an R package so that you can access the data within your R code and directly include it in your downstream analysis. So keep an eye out for that. I briefly want to show you how to access the bulk downloads. So that is available from the main homepage again, going here onto more CEDAR, and then there you can select the database export. This brings you to the bulk download section where we provide the full data sets of all the different sub data sets. For example, you can download just the epitope tab, the antigens, the T cells. So these are all listed here. We provide it in CSV format, TSV format, and Excel format. So you can just click on it and select which one you want to download. For example, this one is the includes all the T cell assay data. Yeah. Okay. To wrap up, here are some key takeaways. CEDAR is a comprehensive curated resource entirely focused on cancer epitope data, and we have a lot of clinical and immunological context. We also link to external resources that you can use information from. We provide flexible queries, and everything that is queried and retrieved can also be downloaded in multiple formats. So hopefully it's very user-friendly. But of course, as Nina mentioned, we always want to hear back from our users to how we can make CEDAR more and improve it to be more user-friendly for you guys. Yeah, and please, you can type in your questions in the Q&A box, and I'll be happy to answer them.
Thank you so much, Zeynep, for that wonderful presentation. I'm going to give Zeynep just a couple of minutes to review the chat. And as she very rightly reminded us, we'd love you to post your questions in the Q&A. Sorry if I just said chat. And we already have a few coming in, so we'd love to see a few more. Before I hand it over to Bjoern, just to reiterate a couple of points. Sorry, Bjoern. The query interface was designed very much on purpose, where the landing page, you can complete about 95% of the queries just by those fields alone. So it's a very powerful and intuitive interface, But as Zeynep said, we're always looking to improve. So if you do have any feedback, let us know. And then once you complete, your first cut of the query, then as you head to the results page, there are even more filter options for you to further refine your query. So that's one of the key features of the database that the team has been working on over the last few years with the learnings from the IEDB. So hopefully you can see that as you use it as well. Start broad and then funnel down into some more details. Okay, so another just quick point was that I did have a user also confirm that there was something a little bit funny with Zeynep, where you were hovering over things. It was not aligning on our end. So I apologize to the attendees. If probably a Zoom glitch, but if that, yeah, thank you to one of our attendees who confirmed that as well. Not an issue. I just wanted to acknowledge it in case anyone else had noticed it. And now, sorry, Bjoern, I will hand it over to you to answer the first question.
Yeah, I had some time to look at stuff as it was coming in. So the first question is, do epitope inclusion criteria differ between IEDB and CEDAR? Will all epitopes in CEDAR be present in IEDB and vice versa, but with expanded cancer metadata available in CEDAR? So the database in which the information is stored for CEDAR and IEDB is completely shared. So anything that is being curated for CEDAR also is accessible in the IEDB. And actually the fields that are used are also exactly identical. So we're using one query interface. The difference is really, one database to store the information. The difference is really not the data, but the query accessibility. So say in the IEDB, you don't have a field where you can just say, let's query for neoepitopes or so. So we capture them the same way in the IEDB, but what is immediately accessible for querying and what is highlighted in the reporting is customized for the cancer community. So that's been our overall thing. And then, yes, if you go into the IEDB and run a query for, say, whatever, give me all epitopes from that are derived from human proteins, you will find some cancer ones and some autoimmune ones and some transplant ones or so, and they'll all be side by side. But if you're really interested in cancer, I would highly recommend to go through the CEDAR interface and so not to be overwhelmed with other data that's available. Zeynep?
So the question is, how does the accuracy of CEDAR data can be verified? So as Alex mentioned in the introduction, we are a neutral database. We just curate the data from the literature, but we provide all the details necessary. So you can, if you want to be sure about the experiments that we use, for example, you can use the PubMed ID, the reference information that we provide, and go back, look at the reference, read the paper, contact the author. So that's up to the user to decide how much they trust the data. So we curate the data from peer-reviewed literature. So we assume that it has been gone through some scrutiny. Yeah, and the rest is up to the user. Bjoern?
Thanks, Zeynep. The next question I have is for query results listing epitopes on the antigens. Could the amino acid position of linear epitopes be included in the tabular view in the future? So on the epitope tab that you're querying for, we are kind of purposefully not listing that because there's one of the confusions can often be that actually there is not an exact match of the epitope in the source antigen that we are listing. Obviously there's like different variations in human proteomes or so, and it can be sometimes hard to identify the exact positions. We do an alignment to the parent protein, and we could include it. So far, this is best accessible in the Immunome browser, where all the peptide sequences and their alignment is presented. And Zeynep, correct me. Are we going to show the Immunome browser at some point? No.
I showed it.
Oh, sorry. Yeah. Okay. Awesome.
Yeah,
So that
briefly,
is
though.
yeah. So that would be the best way of getting at it rather than in the main tab, and there you can really see the potential sequence differences there as well. Well, it's a good suggestion. And for cancer, we might be able to do it. The problem I'm describing is much more acute for the IEDB, where for say sequences in, say, hemagglutinin sequences in influenza A. They're so variable. The positions are really hard to pin down in a general sense. Go ahead, Zeynep.
Oh, I lost the question. So the question was, how do I search for T-cell receptors that recognize class 1 neoepitopes? Maybe I can briefly share the CEDAR website. So I think for that, you can deselect these to just keep the neoantigen, keep the T-cell, the TCR here, and then the class 1. And in this way you can, and if you can, of course, add more details, but that should give you the information that you're looking for. And also you can bulk download all the TCR data that we have, and you just use the Excel table or the CSV table to filter it down further to your needs. Bjoern?
The next question I have here is, can you explain what the difference is between cancer-associated antigens and the other antigens from the same reference in the epitope source box? So actually, I think I'm going to screen share as well. That might make it easier to follow that question. So here is, oops, that was wrong. It's about this field. So essentially, our curation approach is, if there is a paper that has cancer epitope in it, we consider the whole paper cancer relevant. But in that paper, they might be including, say, influenza epitopes as a control that are being tested for recognition by cells from cancer patients to see, is there an overall functional T-cell response in these patients. And so if you click this, then you're going to get these other epitopes back. And so the goal here is to allow for somebody who wants to have, it's like, I see in this paper there's a table, and why do you not return to me this data? That's the way to get the data back. But if you really care only about the cancer-specific epitopes, you should leave this unchecked. That's why it's unchecked by default. I hope that was explanatory.
Next question is, if I want to use the CEDAR data in my publication, should I cite the CEDAR resource? So I believe that should be in the package that Nina posted in the beginning. And we have several CEDAR papers announcing the database and describing how it works. I guess most prominently is the NAR database issue paper, where we show the database in detail, and that's the paper that can be cited. And of course, if you use a specific data from a specific paper that you found in CEDAR, you can also cite that paper using the PubMed IDs that we provide in CEDAR.
Thank you, Zeynep. Next question I have is, most companies interested in CEDAR probably need to keep human privacy. Can they be assured that there's no trackability of human patients? So the queries that we have on CEDAR are essentially queries against public published data. So anything that is in journal articles, that is what we have in CEDAR. So there shouldn't be any human privacy violations, hopefully, in published journal articles. And also the kind of data we capture doesn't include, say, full human genome sequencing data or anything like that. So we don't think that there's going to be really an issue with human privacy. Now, there's other things like the tools that we're going to talk about later. So if you have like a large data set that is actually has like data that is that you shouldn't be sharing in the first place, then maybe also you shouldn't be running a public server like the CEDAR servers to analyze that data. And in those cases, we would recommend to download and work behind your firewall with this data, which can be done for companies also when we use the commercial licensing for the CEDAR tools. But for the data itself, there should be no human privacy concerns at all. Thanks, Zeynep.
There are two questions regarding the programmatic access. One is, when is the programmatic access expected to be available? And if we run a query on IEDB using an API focused on the CEDAR interface type query elements, will we get the same metadata? So to answer the first question, as I said, the API, so we have an IEDB API, which is very comprehensive, but we wanted to make it more cancer specific and also include all the fields that we have available in CEDAR. So that should be available maybe in a month or two. So it's almost ready. And then a question about, so you can use, of course, the IEDB as well to get some cancer data by using, for example, the disease filter. But as I said, for example, there it will not be possible to search for like new antigens or specific mutations or anything, and that we made available in the CEDAR API. So I would recommend waiting and using that one. Yeah.
Thank you, Zeynep. The last question I have here is, you mainly describe T cell and B cell epitopes. Can you query for NK cell receptors and cancer-specific ligands in CEDAR? And the answer is easy, it's no. We are only talking about adaptive immune responses defined by having adaptive immune receptors doing the recognition, so either T cell or B cell receptors. That doesn't at all mean that we don't think NK recognition is important. And it's just like outside of our primary scope. And so unfortunately, you cannot do that. I would actually be curious if there's a good resource for that, and it would be kind of natural to expand CEDAR to capture something like this. But currently, it's definitely not within our scope. And I think that answered all the open questions that I had here. Unless there's anything else, we can take a bit of a break, bio break. Nina, sorry, you're taking this.
That sounds great. The only thing I wanted to reiterate was to the point of the CEDAR citation. We actually, Zeynep was 100% correct. And if you would like to access it fairly quickly from the CEDAR homepage, in this bar up the top, we have more CEDAR and then citing the cancer epitope database and analysis resource. So if you are using the resource in your paper, we'd love you to cite us. Of course, as Alex mentioned at the start, we are tracking citations to our work to demonstrate that the work that we are doing is impactful to the community. So this is a very quick check for how to cite us as well. But Bjoern is very much correct. We do not have any other questions coming through, and we're running about 7 minutes ahead of schedule. So this is a great time to take a little break. Everyone stretch their legs, check their emails, and we will reconvene in about 7 minutes for the next talk. So thank you, Zeynep, and thank you, Bjoern, for comprehensively going through the CEDAR database, the querying and reporting, and all of our questions. And thank you all. We will reconvene in about 7 minutes.